Since microvascular flow varies between capillaries and changes over a time course of seconds, a method for the continuous quantification of flow across entire fields is needed.
STAFF enables a nearly continuous measurement of flow velocities across entire microscope fields within hours that would otherwise take months to obtain manually.
Although these procedures are straightforward, visual demonstration will significantly help new users navigate STAFF for the first time.
To use TrackEM2 to define the vascular network, first select File, New, and TrackEM2 blank to set up a new blank TrackEM2 project, and select the new folder containing the single image from the movie as the project folder. The TrackEM2 windows will open. Right-click in the main work area and select Import and Import Image. Navigate to the single saved image and select the image. Right-click in the main work area again and select Display and Auto Resize Canvas Layer Set. Then left-click in the main work window. The image will fill the work area. To select the areas in the image that contain the vascular network, in the smaller TrackEM2 window, right-click on Template Anything and select Add New Child and Area List. In the smaller TrackEM2 window, drag Template Anything onto Project Objects, Untitled Project. Then drag Template Anything Area List onto Project Objects, Untitled Projects, Anything. Under Project Objects, Untitled Project,